All right, so today we're going to talk about gene expression in eukaryotes. So I did mention in the previous video that eukaryotes, as usual, are a little bit more complicated than prokaryotes, than bacteria. And really, what's kind of cool about eukaryotes is that all of our control of our genes, so this applies to plants and animals, is involved or controlled by the structure of our genes. So how our chromosomes are structured is actually going to be used a lot to help regulate our genes, okay? So I want to talk about the structure of our genes first of all. So in your chromosomes, this purple stuff, this would be your DNA, okay? And your DNA is wound around these blue molecules that are called histones. So histones are proteins that work like spools, and your DNA is wound around them. So the primary way in which we control whether your genes are turned on, whether they're expressed, or whether they're turned off, which means not expressed, is by controlling whether or not they are physically accessible to RNA polymerase. So if you imagine RNA polymerase is a protein, okay, so it has mass, it takes up a certain amount of space, and it will only fit in certain places. So if we take something like RNA polymerase, okay, if this gene is tucked in here, it's not physically accessible to RNA polymerase, okay? RNA polymerase cannot bind to this gene and it can't transcribe it. So this tightly wound DNA, we would say this is tightly wound. When your DNA is condensed or tightly wound on the histones, it's going to be inaccessible. This molecule called methyl is actually going to give it's a chemical signal that causes your DNA to condense, to spool tightly together. So when your body wants to turn off a gene, it's going to put a bunch of these methyl groups all over the DNA. And as it does that, it signals the histones to bunch tightly together. And because the DNA now is bunched tightly together, the RNA polymerase can't fit onto it, and so these genes won't be transcribed. So wherever we see a bunch of methyl groups, or what we would call methylation, we're going to associate that with genes being turned off. If you compare that to this picture down here, we have a different group down here. These are called acetyl groups. Acetyl groups give a signal to the histone to unwind, to open up, okay, to spread out. And so what that does is we're going to see a bunch of acetyl groups on any gene that is turned on or is being currently expressed. So now if we have our RNA polymerase up here, okay, RNA polymerase could easily come down here and bind to this gene and read it. Okay, so the more loosely wound the DNA is, the more likely it is to be expressed. And so we'll find these acetyl groups. So acetyl groups and loosely wound DNA are going to be found in areas where genes are turned on, and methyl groups and tightly wound DNA are going to be found in regions where the DNA is turned off. Also, you have certain genes which activate other genes. So we discussed this a little bit in chapter 11 when we talked about Hox genes. If you remember, those genes that determine where major bodily structures like arms and legs go, they activate what we would call cascades of genes. So one gene turns on two other genes, which turn on three other genes. It works like a waterfall, right? We get this one gene that turns on a whole series of other genes. That's pretty common in humans, is to get what we would call those cascades of genes that activate other genes. Also in your body, you have hormones that help regulate whether or not genes are turned on or off. So for example, growth hormone, when you are growing, is going to activate genes that cause cells to grow and divide more quickly than they would when you're an adult, right, and you're not currently growing. So there are many hormones that are going to control that gene regulation also. So part of the very base level of what's happening is that DNA and how tightly wound it is on histones, but then there are also other things that contribute. So gene products from other genes activating and deactivating other genes, and then also hormones controlling whether or not those genes are turned on or off. But the whole base method is going to come back to this principle of, is the DNA tightly wound or is the DNA loosely wound? Okay. So even hormones are going to control this by, if they want to turn a gene off, adding methyl groups. If they want to turn a gene on, adding acetyl groups. So we've spent a lot of time talking about inheritance, talking about Mendelian inheritance, talking about incomplete dominance, codominance, talking about dihybrid crosses, okay? So all of those things taken together look at your chromosomes, like the 46 chromosomes that are assigned to you. But every organism also has DNA that is not contained in those major chromosomes. So we're going to talk about a few things that you inherit in your cytoplasm. So the main DNA that you inherit that's not part of your 46 chromosomes is your mitochondrial DNA. So mitochondria are pretty cool. If you remember from when we talked about in chapter 6, talked about different organelles, we said that mitochondria resemble prokaryotes, right? They have a double wall cell structure, which looks like, cell wall, cell membrane that's found in bacteria. And then they also have this chromosome that is looped, not in strands. And so that chromosome resembles a bacterial chromosome. Well, there's this cool thing about your mitochondria where the mitochondria of the sperm actually disintegrate upon fertilization. So every single person on the planet, their mitochondrial DNA should be an exact copy of their mother's mitochondrial DNA. If you've ever taken one of those DNA tests where you kind of swab your mouth and then send it in, like 23andMe or the Ancestry one, or can't remember the big one now. But you're going to send away, right, your DNA, and then they're going to kind of tell you certain things about you. A lot of what that's looking for, especially when they're giving you like a location on where your family might be from, right, that you're, European, or you have African heritage or something like that, and they're identifying it to even particular regions like Spain and Germany or Norway or, Russia or whatever. They're telling you you come from a particular place. What they're tracking there are those mitochondrial mutations over time. Now, the accuracy of that is kind of up for debate. So it's not generally as accurate as they want to make you believe when they sell you one of those tests, but it can tell you certain things about what region of the world your ancestors were from. Another thing that happens in the cytoplasm is this genomic imprinting. So sometimes there's a process that goes on that can inactivate or activate a gene. So if you're a heterozygous individual, you may express a recessive trait if its dominant allele was inactivated. So let me just draw this out to explain to you. Let's say we have a mouse. And that mouse has two different genes for skin color, or for like fur color, okay? And we would say that brown fur is dominant to yellow fur. So I'm just giving you two different letters to illustrate this. We could use big B, little b, okay? Actually, let's just do that so it's not confusing. So brown would be dominant to yellow, okay? So now this mouse is a heterozygote, so he should appear with brown fur, right? But let's say that imprinting takes place. Now what imprinting does is it inactivates one chromosome. So I'm going to put this big black square on the bottom of this one to indicate that it's been inactivated. So now that this X is turned off, it's as if he doesn't have this chromosome. So the only thing affecting his genotype now is this. So where he should have been heterozygous, he should have displayed the dominant trait. This mouse would actually end up with yellow fur. Now, imprinting is something that takes place over the whole body as the egg or sperm cells form. And so it's not something that can control small parts of the body. So it's not like only certain cells would imprint. So the mouse would have some brown and some yellow fur. It's something that's going to take place in the zygote as it's forming. So every single cell in the body is going to have this X inactivated. That's what we would mean by genomic imprinting. It's where a gene, a certain chromosome, gets permanently inactivated. I'm just briefly going to touch on this idea of epigenetics, but this is a really cool idea. I'm going to post a couple of books if you guys are interested in the summer and you want to read about some things that are like truly creepy. You can read about epigenetics. But epigenetics is this idea that your environment actually controls your gene expression, not the other way around. So certain things that you're exposed to as a kid or even as an adult can affect your genetic expression. And so there's been a lot of twin studies that have looked at this idea of epigenetics, but basically identical twins have 99.9% identical DNA. So as they grow up, identical things should happen to them, right? If one gets cancer, the other should get cancer. If one's autistic, the other should be autistic. But what we find is that that often is not the case. Usually, and for example, if you take cancer, only one twin will get it. And so what this leads us to know, the conclusion there is that the environment is having an effect on your genes. And you might say, well, yeah, of course, Mrs. Carvalho, like, of course it's having an effect on your genes. But some of the interesting research that's come out of this is in rats, for example. Rats show affection to their babies by licking them. Okay? And so they can tell if a rat mother is affectionate or non-affectionate by counting how many times or how frequently she licks her babies. And now that might not sound super interesting, but what they did is they looked at the genes of the babies in when they had a mother that didn't show them as much affection. And what happened is that a lot of their genes for controlling anxiety were turned off. So mice who had less affectionate mothers expressed fewer genes that could help them control their anxiety. And so as a result, those mice were more anxious or more stressed, okay? Then the real important part of the experiment was when they took the mice of less affectionate mothers and they took them away at birth and they gave them to a more affectionate mother. And then vice versa. They took the offspring of an affectionate mother and they gave them to a less affectionate mother. Now, what this controls for is genetic basis for that affection, right? So now we've eliminated any genetic bias that there might be. And what this should show, right, is that if it truly is the environment having the influence on those genes that help regulate anxiety, then the mice raised by a low-licking mother, so a mother that shows less affection, should show that same suppression of those genes. And what we found is that that's exactly what happens. So basically how the mice are raised with a certain level of affection can actually affect their ability to express genes that would help them to be less stressed and less anxious later in life. Now, for mice, that may not seem super important, right? But what we're starting to realize is that the same thing is true in humans. So basically, how a lot of us were raised as children can actually have an effect on your genotype later in life. Now, that research, some people really like that research because it's interesting and it could allow us to treat certain things. So for example, if we can control the regulation of those genes and we can figure out how to sort of undo damage, we might be able to change, some of those suppressions of genes that we don't want or overexpressions of genes that we don't want. At the same time, a lot of this research is controversial because nobody wants to be told, right, that how you were raised as a child has actually permanently altered your genes, right? That's kind of a depressing thing to hear because there's not really a lot that you can do to change it. And we'd really like to believe that how you were raised, right, doesn't completely affect the rest of your life. And no one's saying that, that you couldn't overcome that, but there may actually be genetic differences that result from childhood, depending on chemicals you were exposed to, ways that you were raised, and a variety of other factors, okay? So epigenetics is this really interesting idea. It's just this newly emerging field sort of in the last 5 or so years that talks about how our genes are controlled by the environment. So like I said, I'll post some books if you guys are interested in that. I think it's super fascinating, so I'd be happy to talk with you guys more about it if you want to chat at some point. But that's just a brief intro to that topic for now. All right, I hate to confuse you with two similar words right after another, but this is just kind of how the book tackles it. So epigenetics and epistasis are really not at all related. I'm not going to go into this too much this time, but what I want to talk about is where there's one gene that can interfere with the expression of another gene. So normally Labrador retrievers have three kinds of coats, right? You can have a black lab, then what we might call a brown or a chocolate lab, and then you can also have a yellow lab. Now there's one gene that controls the coat color, and so being dominant, either heterozygous or homozygous, is going to result in a black lab. Being recessive results in a brown lab. So then you might ask, well, how in the world do you get a yellow lab? And that's where the third gene comes in. So this other gene is going to interfere with the expression of color. So if this other gene, we're going to call it E, if it is either, I can't draw here, erase. So if that gene is either E, so homozygous, or heterozygous dominant, then the color is expressed, okay? So same thing here. If it's little b, big E, these two would get to be expressed. So we would get a black lab here, a black lab here, and a brown lab here. However, let's say we had capital B, capital B. So this should normally be a black lab, but we get the recessive gene here. This gene actually prevents this gene from being expressed. So instead of getting black color, we get no color in the hair, which shows up as a yellow lab. So epistasis is where one gene interferes with the expression of another gene. So a recessive phenotype in this gene here will actually cause yellow coat color that masks the otherwise dominant black or brown color. Okay, so epistasis means there's just one gene that's going to interfere with the expression of another gene. I don't need you to be able to understand this in detail. What I do want you to understand is that there are epistasis means one gene interferes with another gene. All right, two more complexities of inheritance that we're going to talk about before we're done today, and that is genetic anticipation. So genetic anticipation refers to that you have certain genetic repeats in your body. So you have certain sets of DNA that repeat, okay? And that's actually normal. It's normal for us to find repeats in in everybody's DNA. However, there are certain places where an increased number of repeats can be associated with a disease. So one example of a disease where this can happen is called Huntington's disease. So I'm going to show you this picture. Huntington's disease is a really sad disease. It causes muscular degeneration. For those of you that remember like the Ice Bucket Challenge a few years ago that went around for ALS, ALS is a similar disease in that it causes a really slow degeneration of nervous tissue and muscle tissue, and Huntington's disease is the same thing, but it just decreases muscle function slowly. And so what happens is people usually live a normal life until a certain age, and then at that age their muscles begin to degenerate, and so that often means that they lose fine motor skills at a certain point, and then eventually they start to lose the ability to walk, and eventually lose the ability to breathe, and then succumb to the disease. Okay, so this gene, this disease, right, if you found out that you had it, would be pretty devastating. And we have a genetic way of sequencing this. It does tend to be handed down in families, but we can not only tell if you have Huntington's disease, we could actually tell you how severe it would be. Okay, so if you look at this example here, this person here has a normal number of repeats. Okay, so just 10 to 26 repeats would be healthy. However, here we have somebody, Huntington's disease is going to have 37 to 80 repeats. Now, what this means, how we can sort of detect how bad the disease is the more repeats you have, the earlier the onset of the Huntington's disease symptoms is going to be. So if you had only 37 repeats, you might live until you were 50 before Huntington's disease symptoms started to set in. If you have 80 repeats, they could start to set in as early as 20, okay? So this is kind of opening up the importance of genetic testing. You know, if you could find out if you had Huntington's disease, which right now we do, right? We have the genetic testing to be able to do this. It does raise some ethical questions, though, that you should also be thinking about, right? Which is that if you could know about Huntington's disease and that you had it, and you could also know at what point in your life you would most likely be affected. Now, there's not 100% chance that the doctors are going to be right? They're always guessing. You know, it could be 20, they could say 20 and it could not hit you until you're 25. So I guess the first ethical question that I would want you guys to be thinking about, and I don't necessarily have an answer to these questions. The first one would be, if you wanted to find out, do you think you should be able to find out? And how would that information help you, right? You know, do you think it would be a good thing to know that you were going to have this disease so that you could prep and plan for it? Do you think that that might negatively affect your outlook on life? Basically, you know, consider it from a personal angle. But then the other thing we always have to consider whenever we ask if we should make testing like this available to people is, how are we going to protect those results? So if you are able to get genetic testing, who do you want to have access to that? You know, should your partner have access to that? Should your children have access to that? You know, should your insurance company or employer have access to that? That's certainly something that, like, many people would not want their employer to know, right? Certainly not something you probably want your insurance company to know, because what are they going to do? They're probably going to try to charge you more money now, right? Because they know you're going to cost them more money later. So anytime we're looking at genetic testing, and you know, it's very easy to jump in and see how good it is and how helpful it is and how much information it provides us with. But you also have to ask key questions about how are we going to protect that data and who gets to see that data? And how are we going to help people that have gotten these, you know, really depressing diagnoses from genetic treatment? So all of those things need to be kind of holistically considered whenever we're looking at a problem like this. All right, last thing, we're going to kind of shift gears from what I was just talking about, and that is to this really important thing called transposons. And I think I share this because sometimes really important things can come from really boring research. So this is Barbara McClintock. She actually ended up getting a Nobel Prize for the Nobel Prize in Medicine for the research that I'm going to discuss with you. But the way that she figured this out is by studying corn. So she took ears of corn that look like this, and she counted all the kernels on them, looking for this dark purple color, looking for this light purple color, or light yellow color, sorry, that's not purple. She looked for purple, yellow, and then she also looked for a wrinkled or smooth appearance. And what she was doing is tracking a couple of genes. So the gene that gives purple color in corn is dominant to the gene that gives yellow color, which is recessive. But what she found is that on a lot of these kernels, so you can see them here and here and here, is that they didn't have a uniform purple or yellow color. So now if you have been thinking about this, you might say, oh, well, Mrs. Carvalho, that'd be an example of codominance. And the answer is It's not an example of codominance. So then the question became, how on earth is this happening that we're getting part of the dominant phenotype expressed and part of the recessive phenotype expressed? And so what Barbara McClintock discovered is that there were actually these little fragments here, which she called transposons. They were sometimes called jumping genes, is that these little fragments of DNA could actually leave the chromosome and they could insert somewhere else. And where they left and inserted, they would cause an interruption in the expression of the gene, meaning we get a partial bit of yellow showing. Now, that may not seem super important, right? You might be like, wow, how boring. Counting kernels of corn sounds like my least favorite thing to do. And why would we care whether the kernels of corn are purple or yellow? And I would agree with you, right? It doesn't sound super fun to me either, and I certainly don't care what color they are. But the reason she got a Nobel Prize for this is because it showed how those transposons are able to insert themselves into genes and how they control their insertion at certain places. If you remember when we talked about Bubble Boy and genetic therapy, one of the main things that we had a problem with was that we could not control where genes inserted. So the discovery of transposons was a huge leap for us in doing these gene therapy types of research. And so now we're starting to get some successful clinical trials that are coming back with using either viruses or using these principles that we learned from transposons to actually insert genes into people who are maybe missing certain genes or have a mutated copy of a gene. And so especially with in cancer trials, right, where maybe somebody has a particular gene that is causing their cancer, we can go back in and actually infect the people with a virus where the infectious material has been taken out and replaced with the gene we want to deliver. And we can deliver a new copy of that gene that does not cause the same effects. And so this research was really, really important for getting us to where we are now as far as all of those treatments go. All right, I will see you guys on Zoom to chat about this week's lecture and to answer any questions you have.